Okay, so thank you everyone for coming today. This is the second round of the Endosymbioses webinar series. And so for today, we have Gaurav. I'm going to ask him to introduce himself a bit later. But before I go come into that, I would like also to advertise the one for next week. That's going to be Gabriel, and he's going to be presenting his work on creating novel endosymbiosis, artificial endosymbiosis. So I'm also very excited to hear about it. For the next year, I'm still organizing, inviting people. If you have like names that you would like, feel free also to send me an email. And I would be more than happy to take a name, your names in consideration, make invitations accordingly. And but the talk today of today, I'm very excited to hear about. I read the paper; it's very exciting research. And without more delay, please now the floor is yours. Let me maybe stop sharing my screen and here. Okay.
Hi, yeah, thanks for the invite. It's actually really nice. This is the second one, and I also came last time, and it was really good. Um, Now, let me try to figure out the share. This works?
Yes. Mhm.
And
Yes, it's working.
okay. Okay, cool. Thank you everyone for coming. Um, so I am a PhD student at the University of Mainz now in the group of Hanakoko. And this is some work that I did right before my PhD in the Max Planck Institute for Evolutionary Biology. Um, okay, so this is work I did in the group of Chaitanya Gokhale here, and also it's work with Pete Chupon, who used to be in Münster and now is in Marseille. Um, okay, I was asked by Lucas to give some cultural suggestions. He phrased it as like movies, TV shows, and music. I've chosen to do a bit of all of these things. Um, these two are movies, documentaries-ish, and this one is a band. So I decided to start with this to immediately give away who I am or whatever. Um, so Runasimi is a documentary I watched recently, and it's maybe the best movie I watched this year. And it's about a Peruvian father who, along with his son, is trying to translate the animated movie The Lion King into Quechua, which was first a language spoken like thousands of years ago in the Andes, and only second, like a sock company in Decathlon. Um, and this one, Listers, is a documentary. I think it's completely on YouTube, and it's about these brothers. It got really famous, actually. Maybe some of you watched it already. And it's about these brothers that try to do what's called a big year, and it's a year where you try to see as many bird species as you can. So it's a bit like it's about birding, but like incredibly funny also. Um, and yeah, I must have been hit by some like, I don't know, patriotism or whatever. I'm from India. This is a band from India, Peter Cat Recording Company, and they're just really good, like really unique in some ways. Um, okay, so yeah, these are the things I thought everyone should watch/listen to. Um, okay, so now the science. This is going to be a talk about the evolution of endosymbiosis, especially in terms of what I've called dependence and cohesion. Um, so to motivate all of this a bit, I'll start with, maybe the most well-known example of the mitochondria and the host. And now, as we know them now, the mitochondria and the host are completely dependent on each other, and their reproduction is synchronized. So, there's some cell cycle compatibility or whatever you want to call it, but it's synchronized reproduction. Um, so this is some paradigmatic kind of example, but there's also really new ones that we keep finding. This is one that was found just last year. And it's a nitrogen-fixing organelle derived from an endosymbiont in an alkyl species. I try to keep this the newest example that I know about. So if there's newer ones since 2024, I would love to know. Um, but to motivate what I mean by dependence and cohesion, let's think about this tube worm. And this is a tube worm which has an endosymbiont, and the host, the worm, is obligately dependent on the endosymbiont, but the symbiont not on the host. But even though it's obligately dependent on the symbiont, juvenile worms need to acquire horizontally the symbiont every generation from the environment. So there's sort of two things that endosymbiosis can vary in, like the dependence, which can also be asymmetric, different from the host and symbiont side, and also this reproductive cohesion, like are they synchronized or not. And the whole talk today is going to be about placing endosymbiosis in this sort of plane. One axis, the degree of dependence, and the other axis, reproductive cohesion, and then think about evolutionary trajectories in this plane. Okay. Um, so the same plane from before, I'm going to make a definition first. I'm going to say that an evolutionary transition in individuality has taken place only if free living hosts and symbionts go from the bottom left of this plane, so no dependence and no asynchronous reproduction, me and a stick insect from outside my door, to the top right of this plane, like complete dependence and complete synchronized reproduction. So this is going to be a theory talk. All the results are going to be based on a mathematical model that I will introduce. But to like conceptualize these quantities, dependence and cohesion, I'm going to introduce two trait pairs. So the first trait pair, I'm going to call it the obligacy. It's just how obligate the host is on the symbiont. That's the host obligacy. How obligate the symbiont is on the host. That's the symbiont obligacy. Yeah, I hope these words stick, but they were convenient to use. Um, this is one trait pair, and they both will affect the mutual dependence. Second trait pair, I'm calling the adhesions, so alphas, and these affect how often the host and symbiont stay together as a collective and reproduce synchronously versus split and become free living host and symbiont. Okay, so two trait pairs. The omegas affect dependence, the alphas, additions, affect cohesion. The questions are going to be of the form, when can you go from bottom left to top right? And if this happens, and in other cases also, do they go below y equals x, above y equals x, for example, and how does these two, dependence and cohesion, evolve compared to each other? So to quickly motivate the model, I'm not going to go say a lot of maths, and whenever there is, I'll say English also. We're thinking of a population of three kinds of organisms: free living host, H for host, free living symbiont, S for symbiont, and collectives formed by one host and one symbiont. There's a simplification to do analytics. Um, and each of these kinds of organisms can reproduce on their own with some intrinsic growth rates. The F, H, Fs, and Fc are the rates at which host, symbiont, and collective can make one more copy of themselves. And then there's some death rates, and this dynamics is logistic. There's some carrying capacity Ki, and if you leave each type of organism to its own, it'll grow logistically with intrinsic growth rate Fh, carrying capacity Kh, yeah, Fi and Ki. The other kind of process happening in the model is of collective formation and stability. So at rate A, H and S, free living, associate with each other and form a collective. And so this is the rate at which collectives are formed from free living types. And in the opposite direction, collectives can form free living types at these four rates. These rates are sort of denoting two kinds of processes. The BH and BS are the rates at which host and symbiont, while in collective, make free living host and symbiont, right? So there's a collective there's a host that's part of a collective. It makes another host that doesn't have a symbiont with it. This is B. And analogously, think of a collective with one host and one symbiont. If the symbiont in this collective dies, then now it's just a host, free living host. We assume, there's no changes to the host after the symbiont dies, and it's just giving rise to a free living host because of the symbiont death. So these are the Ds, DS and DH. So it's sort of the label is opposite. DS gives an H, but hopefully you don't need to remember this so much. The obligacies will affect these intrinsic growth rates, and they set up a trade-off between the free living growth rates and the collective growth rate.
May I ask a question at this point?
Mm-hmm.
So you always assume that there is a single symbiont in a host, and you don't care about internal populations at all, I assume.
Yeah.
Okay.
Yeah, and the adhesions are going to set up a trade-off between these rates of association, and so the rates at which collectives are formed from free living and the rate at which collectives give rise to free living. It's a trade-off between these rates and the rate of collective reproduction, so FC. So it's like, do you stay together and reproduce the collective FC, or do you do these processes which give rise to free living? Okay. And these are the functions that are used to map these individual level host and symbiont traits, the omegas and the alphas, to these ecological parameters. And we don't need to talk about them so much, but they satisfy the assumptions that I was saying in words now. And all the trajectories are going to look like this. So here, if I'm letting only the omegas, the obligacies, the dependents evolve, the evolutionary trajectories will be in this plane, 0, 1, square. Okay, so all the results will look like this. Okay, so first we're going to look at the independent evolution of both trait pairs on their own. So fix the other trait pair, let one evolve. And in these, we'll look at, do the host and symbiont evolve differently? And then just let everything evolve, see what happens. Okay, first, the evolution of mutual dependence. And to sort of make this easy to get, so this is the diagram I had before, but as a base case, let's think about just this kind of process. Host and symbiont associate at some rate A, collective dissociates at some rate D. And the scenario when I can write all four of these, like group all four of them and call them one number D, is when they're all identical. So, I mean, it's a really special case, but, we'll go out from there. Simple case. So D, and now we're letting only the obligacies evolve and only the host and symbiont obligacy. Then, so this is what happens. Both of them increase. So we're starting always bottom left. Both of them increase, symbiont faster than host, and symbiont hits its maximum obligacy 1. It can't go above this, sort of, we put this in exogenously into the model. And at 1, the symbiont cannot grow anymore on its own, it can grow only as part of the collective. So it reaches this point, and then the host obligacy stops. And we can show mathematically that there's no evolutionary benefit for the host to either increase or decrease at this point. Under the hood, we were doing the mathematical framework that's called adaptive dynamics, and you can show that the invasion fitness doesn't depend on what the host rate is at this point. Okay, so now.
Yeah, I have a question here. Sorry.
Mm-hmm.
Maybe I missed. So obviously there is some asymmetry between the host and the symbiont that causes why the symbiont evolves faster. What is this? Can you just
actually, thanks. I wanted to say it at this point. So thanks for asking. So I assume I put into the model that the intrinsic growth rate of the symbiont is higher than of the host, and that the carrying capacity of the free living symbiont population is higher than the carrying capacity of the host population. So if I flip these orders, these trajectories would go like this and not like that. I mean, this is obviously I'm putting it in, but it's meant to reflect something that I think is not so controversial. Like there's more symbionts than hosts. Okay. Now this was the base case, and now if we go back
Sorry,
to
I do have a question as a
sorry?
continuation of Gergely's. Can you just step back, please? Yeah, I see that the symbiont grows faster than the host, but the the composition or community or how do you call that, the C, it grows slower than the free living symbiont. So why is there an intention for the symbiont to go in? So
Yeah,
what's the evolutionary benefit for it, if there is any?
yeah, I think the intuition for this is that symbionts aren't just as free living symbionts, they are also present in the collectives, just they can only be present in the collectives. So like the number of symbionts, I think like you should be thinking of them as number of symbionts free living plus collective, like it's a combination. And so it's not like there's no benefit for a symbiont to let go of free living. If there's a stable enough collective and the symbiont can be part of the stable enough collective, there is a benefit to going there.
Mm-hmm. But is there a benefit for the host, or you're just
Yeah.
simply not
Yeah,
caring
yeah.
about it?
No, I'm assuming here that so here for the host there's a clear benefit because KC
Mhm.
is bigger than KH. This is easier
Okay,
to
but then it makes the comparison complicated because on one hand you reduce the intrinsic growth rate, but then you increase the carrying capacity. So can you just collapse these properties, like you keep the carrying capacity the same and only play with the growth rate? Because I cannot really fathom the trade-offs between these parameters.
yeah, I
It's
think
just an idea for future.
so it's possible for sure. I would guess that so there's some other results that I have, not for exactly the question you asked, but if I varied the intrinsic growth rate of the collective and the carrying capacity of the collective. So, you can say that the benefit of being in a collective can come in two ways. Either you grow faster or you have a higher carrying capacity. And, if you assume that they're independent and they can be changed independently, you can ask which one matters more for whether these numbers increase or not. And
Exactly.
there we see that the carrying capacity matters a lot more. And I mean, I think the best way to explain this is that the carrying capacity sets more strongly the equilibrium population size. It's not so much set by how fast you get there. Of course, this also matters in a coexistence kind of scenario, but it's set more strongly by the carrying capacity, I would say.
Mm-hmm.
I have a
Okay.
question related.
Mhm.
Um, so it looks like the host is doing the worst when you see the growth rate and the carrying capacity compared to everyone. So I don't understand why they would be the symbiont to evolve obligacy first, because the host is the one doing the worst among everyone. I would expect the host to evolve obligacy first by intuition. I don't know if
Yeah, I haven't thought of it like this. It's a good point. So the way I always convince myself is that there's a benefit. So, okay, given that they increase, there's a benefit for both host and symbiont to invest in collective growth. And it's assumed in the model that any investment more in the collective from either the host or symbiont increases collective growth rate by in sort of the same way. So it's, I mean, we're really modeling only mutualistic endosymbioses. And because of this, it's identical if the host or symbiont invests in the collective, and the symbiont can do this faster because there's just more of them. That there's more of them is an assumption, but more population size, faster mutants come up, faster evolution.
Okay. Maybe just because they have more mutations faster, so they are faster at get there. Okay.
Yeah.
Thank you.
Okay. And now if we go back to this more complicated case, there's many more panels that are going to show up now on the left. So these are results, I should say, that I don't understand so well. So two things can change now. So on the left column is if there's asynchronous deaths on, but asynchronous births off. So recall DH, and DS are the rates at which hosts die while in collective and symbionts die while in collective and give rise to a free living other one. And BH and BS are the rates at which a host while in collective can give rise to a free living host, or vice versa. Not vice versa, same thing for S. Now, on the left, the Ds are on, asynchronous deaths are happening, asynchronous births are not taking place. And is only two cases I'm going to consider here. Both the Ds are equal to 1, both Bs are equal to 0, or both Bs are equal to 1, both Ds are equal to 0. There's obviously many ways to play with this, but we haven't done this yet. So there's two things that can change in these trajectories. One is that when asynchronous deaths are taking place, the trajectory doesn't increase from 0, 0, but after you pass some threshold, high enough initial obligacies, it does increase and it looks qualitatively the same, with the symbiont increasing faster than the host until the symbiont hits its maximum obligacy value, and then the host needs to make, in quotes, like big quotes, a decision of should I invest more now, given that the symbiont is at maximum obligacy, or less. And if asynchronous deaths are taking place, the host evolves to invest everything in the collective. If asynchronous births are taking place, the host evolves to invest less and less over time. Right, so two events are, and the bottom panels are just analogs of the bottom panel from the previous slide. So two things can change. One, whether or not it increases at all from 0, 0, but there's a threshold after which it always increases. Two, at the top edge, do you go, once the symbiont has invested maximally, does the host invest more or less than when this happened? So these are the things that can happen. The parameters are the same as before. Okay, so this was mutual dependence. And really quickly, the other trait pair, what happens to this? And now I'm going to have only this structure for everything from now, just to suppress those effects from the previous slide and see what else we can predict with this model. So same setup, start from 0, 0, and the additions increase, symbiont faster than host for the same reason as before. I've assumed the same asymmetry in these population growth parameters, but now the additions, these traits, go high and then drift along this neutral ridge. And the reason there's this ridge is it's a bit, I would say, because of model assumptions, but it's capturing something real. And this thing is becomes clear when we see at the equilibrium population sizes at each of these traits. So these are the trait values that are underlying this square, and I go to each trait combination and calculate equilibrium population size. That would happen if these were the traits, only traits in the population. And the relevant fact is that these change non-monotonically as you go from bottom left to top right. And the reason is because, so the processes that the additions are changing is, one, the rate at which collectives dissociate to form free living, and two, the rate at which collectives make more of themselves. And so from the perspective of a collective, what's good for this collective is to either dissociate less for its population size or make more of itself. These are the two ways in which the number can go up of the collective. And what's captured by this non-monotonic increase is that initially it's good to invest more in collective reproduction and get this benefit of the dissociation fueled coming back to the free living compartments. So host and symbiont invest in collective reproduction, and at low values of the additions, there's really high dissociation. And so there's a flow back because of dissociation to free living. And this is why they increase to begin with. But at some point, this trade off balances so that it's not good anymore to increase past some value. And the value here is set by these parameters. And you can ask, will it ever go to 1, 1? Will the additions ever go to 1, 1? And they do. I mean, this ridge just keeps going closer to 1, 1, the higher you increase the collective carrying capacity. And at some point, it's close enough to 1, 1. But the point is that they get closer to 1, 1, the higher the collective carrying capacity is. So the fact that there is a ridge, I would say this neutral ridge of evolutionary stable strategies, I would say this would change with more specific D and H and BH, all of this complexity. But here, this result is just saying that there's some balance between the rate of dissociation and the rate of making more collectives. And I think this is the qualitative takeaway from this. Okay, and I can go through this quickly because we talked about it a bit. And this slide was just meant to say that there's all these asymmetries in observed endosymbiosis. You know, it's always the endosymbiont that has a small genome and not the host, you know, relatively speaking. And this was interesting to me, you know, why is it always the endosymbiont? And I think like a really simple way to explain it is that if it's in both the host and the symbiont's interest to invest in the collective, then the symbiont, by the fact that there's more symbionts and so there's faster mutation, faster evolution, the symbiont is just going to invest more in the same time period. So, I mean, there's really complex reasons this could happen. Some of them aren't so complex, actually. There's redundancies. The symbiont is always within the stable environment of the host, and so it can afford to let go of some genes. So, and it's costly to transcribe genes, etc. So there's also these processes. But I think at least some of this asymmetry of, you know, it's always the endosymbionts with the small genome could be because of these asymmetries in the population growth rates. So I wanted to make this point. And lastly, I think I'm already a bit late. I wanted to just go through the what if we let everything evolve. So it's a slightly different model to allow analytics. Yeah.
What was the in one of the slides before you put carrying capacity very, very high? What was the point of that? Sorry, I didn't follow. This
Yeah,
one.
this was just to say that this ridge in this model, it could be also just here, right? Like really close to 0, 0. And then if you believe completely this model only, then you might ask why we see reproductive synchronization in the wild. This model would predict that it never actually goes to 1, 1. If the ridge is really close to the bottom left and it's always drifting along the ridge, these adhesions. And the point of this slide was just to say that if the collective carrying capacity is high and higher and higher and higher, the ridge, the evolutionary stable endpoint of these trajectories is to go to 1, 1, like close to full adhesion, full synchronized reproduction. And
that's kind of interesting because at least people usually agree that for the endosymbiont, the bacteria, they would have like higher carrying capacity outside of the host. I don't necessarily agree with that, but like then would make harder for get a synchronized reproduction if requires the collective to have the highest carrying capacity.
Yeah. So I don't know. So here, of course, KC, the collective carrying capacity, is higher than, where are those numbers? The 200 of the symbiont. But I have not checked if, here Kc is 250, Ks is 200. What if I made Ks also similarly high, like of the same scale, but off by a bit so that it's higher? Like, I don't know yet what would happen, but I should check it though. It's a good point.
I also, sorry, I didn't fully understand the previous graph and the heat map. Yeah, sorry, why do you have the equation on the top of the heat maps? I got a bit confused. What
So,
was the...
sorry, they're not necessarily equations. This is just to say that I'm looking at equilibrium population sizes of the three types,
Okay.
just scaled to their carrying capacity. I don't want,
Okay.
because the carrying capacities are different, I don't want this to play a factor.
Okay. And out of course, the y-axis has like 0, 2, 4, 6, and then okay,
That's
it's
the
8. Okay, I thought it was 0. Sorry.
No, that's actually just um like bad plot making.
No.
Like, it's the indices of the list that contains my discretized version of this axis. So it's actually 0 on the left, 1 on the right, 0 on the bottom, 1 addition on top. So it's the
Okay.
same trait values as this square, just I'm not plotting the evolutionary trajectory. I'm plotting, I'm solving a set of ODEs for each trait value.
Okay. Thank you.
Yeah,
Thank you.
maybe
But what exactly is on the Z scale? So the color scale, it does
it's
show scale population sizes,
it's yeah, I think this is a
or is it
cost
some
of,
intrinsic fitness?
no, it's not intrinsic fitness. I was wondering how much to explain the how I'm modeling this, but if you're familiar, which I think you are, with how like the adaptive dynamics works, um, there's this set of ODEs that describe the ecological dynamics, which is what the populations would do if there was only one trait. And in these heat maps, I am solving this set of ODEs for each possible trait combination in this square.
Hmm.
Zero host addition to one host addition, zero host addition, zero symbiote addition to one symbiote addition. And the question sort of is the trajectory starting from the bottom left of zero both adhesions can go anywhere like this. It cannot increase at all. So what would be the effect on the equilibrium population size attained at each of these trait values of being there, of getting there via some evolution trajectory?
But is it really informative, the equilibrium population size? What I mean is that if none of the parties is going to extinction, then they are all coexisting. So it doesn't really matter what the ratio is, of course, if it's not on a very large scale, but I assume not. So
Yeah.
it would be more informative to see whether the fixed point exists in all these cases or not. The internal fixed point, so the internal equilibrium.
Yeah,
And
it does.
it does, yeah. So what could be a more informative measure? Perhaps the growth rate.
So the informative measure, I would say, is what is in the expression for the invasion fitness. This doesn't always, so I mean, it's true that the invasion fitness does not always have to exactly look like the population sizes. The evolutionary stable direction, sorry, the evolutionary selected direction might be associated to a decrease in population size. This is possible, but in this case, we can show it's not. And this is just a more intuitive picture of, not intuitive, but maybe one of the first questions that I asked, and I would guess someone else might also ask, of what are the population size even like at all of these possible trait values? If I was at complete adhesion from both sides, how many of the host symbiont and collectives would even exist? So this is a bit.
But I wouldn't call it anything close to fitness, because it really is not telling us much about fitness. So we really don't know which one is the winner strategy, do we? I mean, can a mutant invade? That's what we don't know.
Can a mutant invade where?
I mean, if you're in the supposed best strategy, where on the ridge, for example, where you assume that I assume that the population size is the highest. So why would we believe that this is the optimal strategy in any sense, which would equate to equal to good fitness or optimal fitness?
So maybe I don't understand the question. Is it a question about like how I'm actually producing these graphs in the sense
No,
that
not exactly. Maybe I'm not phrasing my question correctly, but I don't see how this image with the scaled population sizes tell us anything about whether this is whether this is a high fitness or an optimal strategy in any sense. To have, for example, high adhesion both for the symbiont and the host. So it's not clear for me why one would say that.
Yeah, so what I will say to this, I think, is that it's not meant to show you exactly why this is the optimal strategy. It's meant to be a demonstration that the population sizes do the same thing as the evolutionary stable state. So there's this ridge of ESSs, and this corresponds to this ridge in the population sizes. Now, why I think is the question you're asking, but this is just meant to show that there is a similar sort of non-monotonicness also in the scale population sizes. And why I would say is because of this trade-off between, like, do you increase dissociation rates? Sorry, decrease, and hence keep more of the free living hosts and symbionts within the collective compartment? Or the other side of this trade-off is to increase collectives making more collectives, this rate.
But correct me if I'm wrong, but there is an inherent asymmetry between host and symbiont in the sense that the internal symbiont does not compete with the free living symbiont, while the collective does compete with the free living host, don't they?
No, they don't.
No one competes with no one else.
Yeah, they don't. This I think is a simplification that I don't want to have. Like, this is one of the first things I would drop when I would do more work on this, but they don't compete. Yeah.
Okay.
Yeah. Do you have any more questions? Although I have to add, since I'm taking questions during the talk is also longer because of this. So it's like I made my talk 25 minutes.
No, it's like it's very nice to be interactive.
I actually really like the questions and trying to answer them also. Okay, then if there's no other questions, then yeah, this is where I was, I think. Yeah, so this is just what if everything's evolving? And this graph, so also because of the slight changes I made to this model to allow analytics, the predictions of this slightly different model, we don't want to trust them for high values of the traits. So this high I haven't defined yet, but we can talk about it if it's confusing. But because of the analytical simplifications, we want to trust it only up to some low value of obligacy. And I haven't made any of this concrete, but just take my word for now. The simplification I'm making is that the collective population can grow exponentially quickly. So there's no carrying capacity or infinite carrying capacity. And this has the effect of destabilizing the whole system in that once these traits become high enough, the population sizes all go to infinity. And this is why so maybe more concretely, this internal fixed point stops existing, and then we can't do adaptive dynamics on this in the same way anymore. This is why we want to trust it only for low enough values. So now I've restricted all of the traits, the joint trajectory of all of these traits, to only what's happening within the host here. So host obligacy, host adhesion, both increase like before. Obligacy faster than adhesion. Symbiont, only symbiont now. Same thing, both increase. It doesn't stop weirdly like the host because it's increasing faster than the host. And this has some effects under the hood, but it's still the same exponentially growing model. But again, the symbiont obligacy increases, adhesion increases, obligacy faster than the host. And if I combine both of these two and ask, does mutual dependence increase faster? Like going back to the very first conceptual plane I showed in the beginning, does mutual dependence increase faster or reproductive cohesion? And here I measure mutual dependence as the product of the obligacies and reproductive cohesion as the product of the alphas, the additions. And then just do the same plots, but instead plot not each trait on its own, but the products. And here, so they both increase dependence and cohesion, but dependence faster than cohesion. And in words, again, this result is showing that if the trait pairs that control dependence and synchronized reproduction aren't correlated, we expect that dependence evolves faster than cohesion under these assumptions. Now, again, why? It's because of these mappings that I didn't talk about before. I'm going to talk about them a little more now. So the two trait pairs that are having different effects are these obligacies, the dependencies, and the additions. So all four numbers, traits, have the same effect on the collective reproduction. Like you increase any of them by 0.05, will have the same effect on the intrinsic growth rate of the collective. However, the growth rate, the effect on the free living types, we assume the effect of the intrinsic growth rate of the host does not depend on the obligacy of the symbiont. So how much ever the symbiont invests in the collective, this doesn't affect how well the host grows on its own. This is an assumption. I've put this in, and it's meant to caricature like the fact that host and symbiont in the system, we want them to interact only at really close proximity within the endosymbiosis, and also the other way around. But the dissociation of collectives forming free living hosts and symbionts again, this I've put into the model that it's a product, it's affected by the product of additions. So it's some kind of coordination process. Like there's more dissociation when both of the additions are less. And this is because I'm thinking of dissociation as sort of like a together process. And because of this, a slightly lower value of adhesion is always modulated by this addition of the other one, which is a number less than 1. So it's always, the effect is lesser. And so this is why we're seeing this effect. If the effect of investing more in how dependent you are in the collective can affect the host or symbiont independent of the other one. If these are separate effects, but the association and dissociation are sort of coordinating effects. They happen only with the product of the additions, then we expect this bias. So of course you can change how these functions look, and this trajectory, the bias you can change also which part of the diagonal it is. And there's like a set of predictions that you can make based on this. Okay, so
So
yeah.
for the symbionts, I feel I have a kind of good intuition, like in the sense, if you have higher obligacy, it means that you cannot survive on your own, so does not matter to have higher addition for the symbiont because you cannot survive on your own anymore, so you can only survive inside. For the host, I would expect kind of the same, right? Because if you increase the obligacy, actually, no, actually it'd be the opposite. You would actually try to have more addition in the sense that if I lose my partner, I should find really fast a new partner. Otherwise, I could not survive.
Yeah.
But but in your case, it's kind of like close to diagonal, so it's kind of they should be equally important for the host, right? Does it make?
Yeah, it could make sense. Yeah, I don't know exactly how to respond to that, but I would think that if you sort of cut the symbiont trajectory too early, so not high values of the omega and alpha S, it looks sort of the same. I haven't done any, quantitative things on this, but it looks not so far away from the diagonal for the symbiont either. So maybe it's just that these effects are amplified at higher values of these traits, omega and alpha, and so that's why it goes further away from the diagonal. And the fact that it's amplified more for the symbiont is because I said these population growth rates are faster for the symbiont and the carrying capacity. Um, it's not a satisfying answer, but maybe that's why.
Why for the joint evolution it only goes until not like one? What's the reason for that? Is because
The one top right, you mean?
either the symbiont obligacy or the sorry, either the joint dependence or the reproduction cohesion of both together in the joint evolution, it goes only to the parameter space that is uh, O.4. They're not like
Yeah, so
extrapolate.
this is because it's not a real biological reason. This is again because of the reason because I said that we want to trust the results of this model only up to low trait values.
Okay.
So if the immediate next step would be, we didn't do this because it was like a lot more to figure out. But if I did the same kind of simulation with instead not an exponentially growing collective population, which is what's going on here, but with some carrying capacity, then one of these would go to one. Or, we would see dynamics like all the slides before. But there's also, the question of what if I have these BH and DS and BS and DH, all of these also. And we just wanted to stop here and do the next bit later. But yeah, so all of the real takeaway from this should only be that at early trait values, early dependence and early synchronized addition, sorry, not synchronized, we expect there to be more dependence than cohesion. Not, I don't know if this would be true also later. So yeah.
Oh, can you also explain why the growth rate of the collective includes the adhesion values? Because to me it seems a little bit counterintuitive that the death rate has or the dissociation rate has these values and the growth rate also has these values, because then it's almost as if they were squared, because it's reproducing faster and then it's also dissociating faster, right?
Yeah. Um, so it's a good point. It's not completely realistic. I think the right improvement on this would be to take into account not just one number D, but all of those DHBS, all of those, and then say, so I think there's going to be an asymmetry in a slightly different way. So there's these births of free-living host and symbiont from the collective, and also deaths of host and symbiont while in collective, giving rise to free living. And I think what the adhesions will affect is, which one? Yeah, the rate at which free living hosts and symbionts give birth to free living, the rate at which hosts and symbionts while in collective give birth to free living. So like asynchronous births or something.
Oh,
So I
yeah.
think this actually will trade off with FC. And it's a bit unrealistic because I'm calling all of them by one number D. So yeah, there's a lot of letters now, but the rate at which you make free living offspring as opposed to a collective offspring while in the collective, I think these two should trade off with each other.
Yeah, I think that makes sense.
But yeah, I agree here it's a bit not perfect. Does this answer the question though? Sorry.
Yeah, I think so. I'm still processing.
Yeah. Yeah, maybe I should have had more words. But yeah, thanks for the question. Okay. So really quickly, I think forgetting all the details of the specifics, like one of the more qualitative things this shows is that as soon as, even if there's the existence of a higher level of selection, so collectives can make more collectives, this doesn't imply that an evolutionary transition is always going to happen. It's a bit trivial in some ways, but I think it's still interesting to say. And when does our model suggest it can happen? High additions can evolve, close to full additions can evolve with high collective carrying capacity, and high dependence can evolve when number of asynchronous births decreases as dependence increases. So the births need to be high at the beginning, low at the end. This is sort of how I'm choosing to summarize this result from before. Third, we can explain asymmetries just by accepting a simple fact, which is true in some cases, maybe not all, that there are going to be differences between hosts and symbiont population growth parameters. And lastly, we shouldn't expect dependence and cohesion to evolve at the same rate. One could be more than the other. I would say the most realistic assumptions lead to dependence being faster than cohesion. And this is what I would look for. I think this is a testable prediction, and we can actually go out and test endosymbiosis, do an evolution experiment, etc. Okay, yeah. Oh, I had the slide. I just really want someone to do experiments on this. Like, I think, like, to really have an evolution experiment with somehow selecting for collective reproduction and measuring the free living and collective growth rates and doing all of this systematically, as far as I know, no one's done this, and this would be really so cool. Yeah. Okay, this is it. Thanks for listening and the questions.
Thank you very much. I really enjoyed it. I think it's a very nice and clear-cut model that has a very high potential to provide further insights on the evolutionary transitions of multi-level selection scenarios. Though, and unfortunately, I don't have much time because I have to run for the kindergarten. But I do have one question. How much do your results depend on the actual choice of parameters you had for the different populations? The original question that we asked very early on. So what happens if you change the symmetry of the symbiont and host population? And for example, it's more beneficial for the symbiont to make a free living life. So what would be the incentive at that case? I mean, you don't have to answer that now because clearly it would require more simulations, but it would certainly be interesting to see how these would affect the results.
Yeah, I mean, maybe one quick thing to say is that in all of what I was presenting, there's no description of the fact that the symbiont is inside the host and not the other way around. It's only a description of which one can grow faster in isolation. And now I've chosen to impose that, I could have just as easily called the host as the one that's growing faster and having a higher carrying capacity. And this is a bit like interpretational, like do we think that symbionts have more often a higher carrying capacity or the host? I'm of the opinion symbionts. But I think for all of these results, if you think the host grows faster and has a higher carrying capacity, just switch H and S in everything I said.
I see. Perhaps then it would be useful to depict them as such. So not like one in the other, but they are just simply active symbionts.
Good point,
Because
yeah.
then it would be very clear that the situation is isomorphic if you exchange the partnership or the roles. Yeah, that makes much sense. Yeah. Okay,
Yeah.
thank you very much.
Thank you for your comments.
I think maybe Jakob had a comment, or maybe not. I saw you unmuting yourself.
Oh no, I found it very interesting, but no further questions.
Thanks.
Any further questions for anyone else? No? So, yes.
Yeah, just a suggestion that you might just want to send around your paper for us to read. Of course, we can download it, but I will forget it for tomorrow.
I could send it to
I can send
Lucas
to everyone.
and
Yeah. I have the email for everyone, and I can also send in the Symbiosis webinar group that I create. You can subscribe. So if you are subscribed there, you can also receive the paper. Yeah, but beyond that, thanks so much for the talk today. It was really exciting. It was really cool. Yeah, it was a lot of fun. It was much more fun than I expected.
Yeah. Thanks for the talk.
Yeah. So.
Yeah, thanks for coming.
And yeah, and that's all then. And next week we will have it's going to be the same day of the week and the same time, and it's going to be on experimental. So maybe you can try to pitch your project to him. Yeah, maybe it'll be cool.
I would love that. I can't make it though, unfortunately, but yeah.
Okay. Have a good day, everyone.
Thank you all. Bye bye.
Thank you. Bye.
Bye.
See you.
You.